In this video, we will discuss about how they find out a replacement of the promoter gene, that is PDC, PD1, that gives a more systematic toxic effect in terms of the weight loss that we have discussed in the previous video. So in order to find out the genes that helps in tumor specific transgene expressions, what they did is they compared the genes that are expressed in the activated T cells in the spleen and in the tumor. And what they are able to see is that there are many genes that are overexpressed in the tumors compared to the spleen. And out of these genes that are overexpressed, they find out 27 best candidate based on their differential expressions compared to the spleen. And out of these 27 genes, they are able to see that 12 of them has more differential expressions compared to their control case, that is PDC D1. That the 12 genes that are highly expressed are shown with orange, and the PDC P1 genes shown in the black. The rest of the transgenes, but they are expressed lower compared to the PDC P1, are shown in the blue. Thereafter, what they did is they prepared the individual knockout CAR T cells and check whether the activated form of those CAR T cells affect the cytokines formations or not. What they are able to see is that there is a differential expression of the cytokines in individual knockout, and out of this they selected the top 6 candidate CAR T, top 6 genes that expressed the cytokines. However, they eliminate the AK4 and one more gene because of their very low expressions of the cytokines, and they include TNF AIP3 because of its high expressions of the interleukin 2. And so once they identified these top 6 genes, along with that they use the TNF API3 and PDC P1. So in total they have 8 genes. So in these 8 genes, first they did the in vitro tumor co-culture with the CAR T cells to find out, and in this in vitro co-culture CAR T cells, they use it with the system that they have discussed in the first figure that we have covered in the previous video. That is they introduced the GFP promoter sequence just downstream of their transgene start codon. So they wanted to see whether those activated CAR T cells will be able to form or express the GFP expressions or not. And what they are able to see, around 4 of them are able to express GFP signals, and those 4 are this NR4A2 and RGS1, RGS16, and this PD1 or this PDC D1 control. And the quantifications are shown in the panel E, where we can see that NR4A2, PD1, RGS16, RG2 has significantly high expressions after 2 hours compared to the 24 hours. So once they identified these 4 candidates that shows high significant expressions between the activated cells and the non-activated CAR T cells, they decided to use these 4 candidates for the in vivo studies. So they did the similar in vivo studies, and they wanted to check in the in vivo studies about their specificity, whether the CAR T cells are expressed near tumors, or whether the CAR T cells with GFP expressions are present only near the tumors, or they are also present near the spleen. So they first introduced the Silai Levis antigen positive mice, and then introduced the Silai Levis anti-Silai Levis CAR T with different knockouts, and then check GFP expressions after 14 days. What they're able to see that apart from this control, PDC-D1, they are able to see high expressions in the this RGS16, and also they are able to see high expressions in this NR4A2. High expressions doesn't mean the high expressions only in the activated form, but they should see a significant difference of the signals between the activated form and between the activated CAR T cells and the non-activated CAR T cells, or near the tumors and near the spleen, because near the spleen the CAR T cells should be non-activated form, because there is no cellular levies or no or minimum cellular levies tumor cells are present. So this difference is shown, can be seen more in the NR4A2 and also in the case the RGS16. And when they quantify this percentage GFP signals in the in vivo studies, and they are able to see that significant difference can be seen in this NR4A2 at the spleen and also at the tumors. And there are other cases also where they can see the significant difference. For example, in the control, that is PDC-D1, also they see the high difference. The RGS16s also they are able to see the high difference. And for this clue, but the total intensity of this GFP is lower compared to the other cases. So thereafter they check it using the difference between the tumors mean fluorescence intensity, GFP mean fluorescence intensity minus spleen minimum spleen mean fluorescence intensity of GFP. And in that case they are able to see that for the case of RGS16 the data is actually significantly higher compared to the control case, that is PDC-D1. So from that they pick up a candidate, this RGS16, and from the GFP expressions they pick up a candidate NR4A2, because NR4A2 has shown more specificity compared to the CLU and also compared to the RGS2. So they pick up two best candidates, one from the one's from the this specificity, and one from the difference in the signals, from high difference in the signals of GFP from tumors and spleen. So these two candidates that they check for the safety, potency, and their further functional studies are being carried out on these two candidates that we will discuss in our subsequent videos. So far, after their in an attempt to find out alternative promoter gene, they come up with alternative promoter gene apart from this PDC-D1 because of its side effect, systematic toxicity. They come up with this NR4A2 and RGS16 gene. We will discuss in the subsequent videos what are the other studies or safety, potency experiments that they did to check whether these promoter genes are good for the tumor specific transgene expressions and has minimum toxicity or systematic toxicity. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.